Are you working with immunofluorescence and you are interested on how to go from this resolution to this resolution? Or from this image of a glomerulus to this image of a glomerulus? Then you are in the right place because this is what I will discuss today. Hi, my name is Julia and I am a PhD candidate at the Veterinary Faculty of UGent. Today, I will talk about how to tackle high background fluorescence and low target expression and how to resolve nanoscale structures in thick tissue sections using FLIM and STED. My work revolves around the Thai receptors in kidney podocytes, and part of my research focuses on localizing these proteins on the podocyte cell. To do that, I use super-resolution stimulated emission depletion microscopy and fluorescence lifetime imaging microscopy in collaboration with the GLIM Core Facility Center. Now, why do I need to use these fancy techniques, you may ask? Because during my experiments, I was faced with certain challenges. I have high background fluorescence. My secondary antibodies bind non-specific to other antibodies and to the tissue. My target protein of interest is very low expressed on my cells. And I want to be able to accurately localize the Thai protein, but because of the diffraction limit, I can't do that with conventional microscopy for structures that are very close to each other. So how do we tackle these challenges? Well, FLIM will be our best friend when it comes to autofluorescence and non-specific binding. Instead, will lend us a hand in the nanoscale structures. The low target expression? A bit of both FLIM and STED will help us here. So what is FLIM and STED? Starting with FLIM, fluorescence lifetime imaging microscopy measures the lifetime of a fluorophore instead of its intensity. Fluorescence lifetime is the time interval between the moment a laser beam excites a molecule until this molecule falls back to ground state and releases a photon. Instead, stimulated emission depletion microscopy is great if we want to visualize single individual molecules that are closer to each other than 200 nanometers, such as proteins on a cell membrane. It works by using a clever way to bypass the diffraction limit by utilizing two lasers, an excitation laser and a depletion laser, overlapped to create the so-called donut effect. In this way, only the molecules inside the donut center are excited at their correct wavelength. And how do we start? Well, before we get to the exciting imaging part, we need to prepare our tissue. I use mouse kidneys that I fixate in formaldehyde and after I embed in a hydrogel solution. I then use a vibrotome to slice 100 to 500 micrometer sections and I clear them in a clearing solution. Next follows an immunolabeling protocol, and I then embed the slices in a fructose solution. Last step is the imaging with either a confocal, a STED, or FLIM system. Let's look at some pictures and see how we can resolve the challenges. How do we tackle autofluorescence? Here you see an image of a kidney glomerulus without any fluorescent probes, captured with FLIM. You can see that there is signal present, very bright from the red blood cells and a bit from the surrounding parenchyma. If we check this signal on a phasor plot, we can observe the cluster corresponding to it. And when we trace it back to the image, we see that the red blood cells are the ones that come back pink. In this case, the parenchyma signal we can exclude by applying certain thresholds post-processing. How about non-specific binding? First, on the left, you have an intensity image. And by looking at it, one might conclude that there is only one fluorescent population here. But if we check the FLIM image, we can immediately distinguish 2 to 3 populations marked with blue, shades of green, and orange-red. Why is that? Because lifetime is an intrinsic property of a fluorophore that changes depending on the fluorophore conformation or the microenvironment in which the target molecule is present. Meaning, if our secondary antibody binds non-specific, it will have a slightly different lifetime depending on what and where it binds to. This is exactly what you see here, as my secondary antibody did bind to two different primaries, resulting in two different lifetimes, the green and the blue one. And what about if the secondary antibody binds to the tissue? This sample was only stained with secondary antibody, and by just looking, we see that we have at least 3 different cell populations. We can mark these populations on the phasor plot and trace them back on the image with red. And why is this helpful? Because in the software, we can keep all the areas we marked on our phasor plots for the autofluorescence and the non-specific binding controls. And we can then go to our positively stained sample, and we can now look only at our specific binding, in this case marked in white. So even when overlap is present, as seen in red on the image, we can still separate the signal. How about low target expression? For low targets, we can use STED, because we will be able to better separate the signal that comes from the interest molecules compared to background or other signals. On the right, we have the FLIM image. We immediately gain additional data by using FLIM, and we have an added advantage because lifetime is not dependent on fluorophore concentration, to a certain limit. And in most cases, FLIM will be able to detect very low signal in samples, as observed here. And lastly, nanoscale structures. Proteins are very tiny structures that are found at distances less than 200 nanometers of each other. And because of this, we cannot accurately separate them in conventional microscopy. But with the use of STED, we can now increase the resolution of our images. And you can see here on the far right, my scale bar is 0.1 micrometers, which is 100 nanometers. And by applying post-processing, you can go even lower than this. Good. In conclusion, let's summarize. Can we tackle tissue autofluorescence using FLIM? Yes, we can. Can we separate the non-specific binding of our positive signal? Yes, we can. Like for autofluorescence, by using FLIM, we can mark distinct lifetime populations of our controls and exclude them from our positive stains. Can we visualize low-expressed targets? Yes, we can. FLIM is non-concentration dependent and instead increases our resolution, making low target signal accessible to us. And lastly, can we separate proteins that are closer to each other than 200 nanometers? Yes, again. By using a clever technique developed by Stefan Hell, we can now visualize structures that less than 10 years ago we could only hope to see by means of fluorescence. With all this in mind, I hope I have given you a small insight of how FLIM and STED can help you improve your immunofluorescent experiments and take them to the next level. I thank you for your attention.